template as well for your assignment. So again, it's essentially just a summary, or it's a written assignment of a summary article. The article is titled Effects of Diet Modulated Autologous fecal microbiota transplantation on weight regain. And again, it can be accessed through this link or in the assignment tab on Quercus. And so your goal will be to summarize the research aim, the methods, and the findings. And at the end of the summary, you also need to provide a critical evaluation. So you need to assess like the strengths and the limitations of the research summary and suggest also potential future directions. So where could this research go, what it could lead to, what it means. Yeah. So it's very important to write in your own words. Don't copy anything directly from the research article or from any other research. Use the resources available to you. Obviously, there are quite a few, so use spell check to avoid grammar mistakes and just little errors. And we are here to offer support, but we do recommend that you get started on this early because obviously we also get really busy with assessments around a certain time. And so you don't want to leave it until the last minute, and we'll be more than happy to help. You can email either me directly if you have any personal issues, or Dr. Pinelli, and you can also reach us via Quercus to ask any questions. This assignment is worth 25% of your grade, and it will be based on 75 marks of content. The compliance with the instructions, again, is 15 marks, so you don't want to make any mistakes in not following instructions. The comprehensiveness, and so the flow of your paper is written in your own words. Does it make sense? How you have understood the article and how you've translated this to us will all be marked. And so it's due on Tuesday, November 22nd at 11:59 p.m. Via Quercus submission. You cannot do an email submission. It has to be submitted online. And if you have anything, or if you submit anything later than this date, there will be a 10% reduction of your grade. And each day that it's late, it will just be an additional 10%. So if you're 3 days late, you'll have 30% deducted off. And yeah, so this is the assignment written details for regrading papers. And so you will need to submit a proposal as to why you think you need a regrade. And again, this needs to be typed and formatted using the same summary outline as you used for the assessment. And you need to email explaining why you deserve a regrade. And so the combined average of my grade and Dr. Pinelli's grade is what will be considered, and this will be taken as your final grade. And so your grade, depending on how we mark, may either increase, decrease, or stay the same. And so if you also want to meet with either of us or both of us to discuss the paper or to obtain like certain clarification about how it was graded, you just need to email to request like a meeting time, and we can definitely set up a Zoom meeting. Yeah, and then if you want more information just about like regrading policy, you can find it in the syllabus, and it's also found on like various U of T websites. It's pretty standard across all departments. And that's it for the assignment.
Thank you, Anna. Any questions for Anna?
Yeah.
It's clear. Good. So on Quercus, there are two discussion boards. One is for the assignment specifically, and one is for the course content. So feel free to post any question that you may have in the appropriate board, so that we can answer to you like promptly, and also, you know, many times, as I'm sure you know, like the same question comes up multiple times. So if you put it up there, then it's available, the answer to everyone. You don't have to put your name if you don't feel comfortable with that. You can just post the question, okay? So don't email me or Anna with questions about the assignment. Use the discussion board for that. And but you can use the email if there is a question of more personal nature, of course, right? But yeah, about the content and how to do this and that for the assignment, or about the course content, then please use those discussion boards on Quercus.
Yeah, I'll monitor daily, so your question should be answered quickly.
Yes. Yeah.
Okay? Good. Thank you.
So is everything clear before we go into the material for today's lecture? Thank you, Anna. Yeah, that's fine? Okay. Okay, good. tell you now, some of you may consider really basic. Right? But because you come from so diverse backgrounds and you have taken so many different courses, and I know many of you are not taking microbiology, that's why I'm doing this, right? Also, because with the fermentation of the microorganisms, you may have some recollection from your biochemistry courses, for example, and I'm not going into the details and the reaction and the electric pathway. But I just want to put it there so that you can situate where we are and what we are talking about, and you can really understand where the bacterial metabolites will be interacting with us, where we use them. So that's the purpose of this basic lecture for today. So generally speaking, the microorganisms can be comprises many different groups of different organisms, including both the prokaryotes and the eukaryotes, right? So prokaryotic microorganisms include archaea and bacteria, and we'll talk about both of these groups during this course. So for those who may not remember, I give for granted that you know what bacteria are. Like archaea are also unicellular prokaryotic cells. And they are very particular in terms of their preferred habitat. Like most many different archaeas are very strict anaerobes, and they are found in these hot springs, like in Yellowstone Park, for example, in these hot water springs, or in this very salty and hot environment where many other microorganisms would not be able to thrive. This is where we find many different archaea. And we also find them in the intestinal tract, which I don't know if it surprises you or not, but it's not that friendly as an environment. So these microorganisms that reside in the intestinal tract have adapted to that environment and have developed like the capacity to thrive in this ecosystem. It's difficult, at the same time it's also a place, the intestinal tract, where there is a continuous supply of nutrients. So these microorganisms have learned to use those nutrients that are available to them in there. There are some archaea that are also associated with some of the foods that we eat, especially in their raw form. So a main archaeon that we hear about a lot during this course is called Methanobrevibacter smithii. So this name, you will have to learn it and to remember it. It will come up many times, so at the end it will become easier for you to remember these names. So obviously this gives, like open parentheses, this is a microbiology related course. So there will be a name of bacteria, right? And some of them you will have to remember. Many students are worried about this because they say that these names are complicated. And I know, like, they're not like easy names that belong to our usual daily language. But that's what the course is about. It's about these bacteria and microorganisms, so we need to know the name of what we're talking about. These names, as you will learn, have sometimes have meanings. So if you see the root of the name and you try to, and you understand why they're called like that, then maybe it's easier for you to remember some names. I will not expect you to remember the names of all the microorganisms that I will mention during this course. Obviously, I'm giving the lecture, you're reading papers, you will come across a lot of names and I have to name them. But I will specify which ones you have to remember. Okay? So this is one of them. The Methanobrevibacter smithii, you will have to remember this name. So this archaeon that resides in the intestinal tract of many people, although not all, even though there is still some controversy about this because some researchers think that we have not found it in everyone just because our techniques are not sensitive enough. But at least then we can say that they are present at a detectable level only in about 2/3 of the population, and the remaining 1/3, they may be there, but at very low levels. So anyway, this Methanobrevibacter smithii, as many other archaeon in the gut, is the main methanogen. So it produces methane, CH4, which we will learn why this is important later on, but just wanted to mention that right now already. So bacteria, on the other hand, are also unicellular prokaryotic microorganisms. They can be distinguished by the different structure of their cell wall, as we will see in a few slides. And they can live in association with many other bacteria, but also with archaea. Right? So obviously, the intestinal tract is an example of a site where bacteria and archaea coexist. Right? And this co-occurrence has been, it's a recent discovery. Right? So it has been the importance of this co-occurrence. We haven't figured it out completely yet. Right? But in the past, I would say 5 to 10 years, that we are aware of the importance of the relationship between archaea and bacteria, which gives me an opportunity to open another parenthesis for something that you will hear a lot in this course. You will hear me saying, oh, this we don't know for sure yet, and this has not completely been elucidated until now. That's because this field of research is very, very new. One reason it is very new is because until 15, 20 years ago, we didn't have even the techniques to study these complex bacterial and prokaryotic communities everywhere, not only in the intestinal tract. So it is only during the past 15 to 20 years that we have produced the knowledge that we are discussing in this course. To you, maybe 15 years seems a lot of time, and I realize that in your lifespan, like with your denominator, 15 years and 20 years is a lot. But, you know, to produce the knowledge, to verify the knowledge, reproduce it and make it usable in the field of research, like this is not a super long time. So there's still a lot that we have to understand and learn about the microbiomes, and including the gut microbiome. Right? So you will hear me say this a lot, and maybe sometimes I will not have answers to some of your questions because those answers have not been found yet. Okay, so let's go back to this. So an example of co-occurrence between a bacterium and an archaeon, which may be relevant to human health, is between these Metallographic metii and this bacterium which is called Christensenella minuta. Don't have to remember this name, but you have to remember the concept. I just want to give you an example of why this co-occurrence may be important. So when these prokaryotes live within a community, their metabolism, as it may seem obvious, adapts to the fact that they are not living on their own in a pure culture like we can have in a tube in a lab, like a tube that looks like this, for example. But they are in a complex community with many different different types of bacteria, and they live together. So one thing that may happen is that they develop cross-feeding mechanisms. So that the metabolites that are produced by one of them are necessary for the growth of the other one, or they modify the metabolism of the other one. Some other times, as we will see later on, some metabolites are detrimental to other microorganisms. Right? So the fact that they live together means that they have to thrive in an environment where now there are metabolites that are being produced which they may not be exposed if they were living on their own in a pure culture, right? So what happens between Christensenella minuta and Metallographic metii is that when they are together, there is a situation of syntropy that occurs. So syntropy, the definition is here, refers to processes of cross-feeding and reliance of one organisms on nutritional factors being produced by another. Okay? So what happens when Christensenella and Metallographic metii are together is that the bacterium, the Christensenella, produces, among its metabolites, produces hydrogen. And hydrogen is what the archaeon, Metallographic metii, needs for its methanogenesis reaction to produce methane. So it is in the presence of this bacterium. This bacterium provides a substrate for the archaeon. And interestingly, they were able to show in this research published in this paper that in the other direction, the metabolism of the bacterium Christensen-aminote is also altered. So the metabolites that it produced in the presence of the archaeon are different as compared to those that it produces when it's on its own. And because these metabolites are short-chain fatty acids, which we will hear again in a few minutes, and then a lot during the course, which have a role in intestinal health and metabolic health, and they also have been related to obesity and the metabolic syndrome. So because the archaeon alters the production of the short-chain fatty acid by the bacterium, so this may be a mechanisms by which these two prokaryotes in the gut may affect the metabolism of the host. So we will discuss these relationships much more in detail throughout the course. But it is good that you start realizing and having some hints already now, so that then it's easier to put things together as we progress. Okay. So so this is a syntropic interaction, as I was saying, and in this example of syntropy, the Christensenella supports methanobrevibus vitae through production of hydrogen. Okay, the other three classes of microorganisms, won't spend too much time on this. This slide is here for completeness, but we will not discuss this much during our course. So there is the algae that are photosynthetic and eukaryotic, the protozoa that are non-photosynthetic eukaryotic, also found in many different environments, and also the intestinal tract, and fungi that are non-photosynthetic eukaryotes that include molds, yeast, and mushroom. And we discussed this next week. You'll see, for example, some yeasts, how they can be used in the food industry. And when else? Yeah, I think that's most of what we will hear about yeasts in this course. So briefly about bacteria, I said they are unicellular microorganisms without a nucleus shaped as spheres or autospiral. These are just some examples. So for example, this Lactobacillus acidophilus, Bifidobacterium longum. You may have heard these names if you go to the grocery stores and look at this product that they are sold as probiotics and read on the label for these. Sometimes they contain some strains from these two species. Streptococcus thermophilus is a bacterium that is utilized. Some of the strains can also be probiotics, but it's utilized in the production of some of the dairy foods. So you will hear a lot about those next week. How do they reproduce? So the bacteria mainly reproduce by binary fission, which is an asexual process and which consists of an increase in the cellular structure and components, and then replication segregation of the bacterial DNA. There is a septum that forms, that is shown there, and then the cell divides into two daughter cells. There are ways for measuring the generation time, which is the time that is needed for a number of bacterial cells to double. So we can use many techniques, and I'm going to mention a few because we may discuss some data later in the course or in the papers that you read in the literature where they provide measurements that were obtained with these techniques. So I want to make sure that you know what we are talking about. So if we want to count bacterial cells, which can be in a food product, maybe we want to know how many bacteria there are in the yogurt that is being sold, because, for example, there needs to be a certain number to meet the regulations for the law for selling that product. Or we want to know how many, lactobacilli or clostridia there are in the stool sample of a certain individual who has an inflammatory bowel disease versus a healthy person. So there are many techniques that we can use to produce this number. So the most direct one and basic is the direct microscopic count, right? So you prepare like a microscope slide where you put some of your diluted sample and you count the bacteria under the microscope. It's relatively easy, but you cannot distinguish from living and non-living cells. So it depends on what your outcomes and what your needs are for when you enumerate these cells. So if you are only interested in the number of cells that are alive, so I'm going to use the example of a probiotic product again. So a probiotic product needs to have, to be able to sell it as a probiotic, it needs to have a certain number of live cells at the end of the shelf life, so the expiry date that you read on that yogurt product. So if we want to determine if that's true, if there is compliance, then we need to enumerate the number of live cells in that yogurt, not the total cells, which may include dead and live. So to do that, we use the colony count method. So we use culturing, right? So we dissolve the food products, for example, or the stool, if you want to know how many live E. coli cells there are in a stool sample, right? This can be any product that is just dissolved into a solution. We prepare serial dilution, and the last dilution, so the less concentrated one, we plate on an agar plate. What is an agar plate? Is like a round container that contains a medium, so a nutritional support for the bacteria that we want to count, which is in solid form because we add agar, which solidifies and provides like a solid support for the bacteria to grow. So each bacterial cell growing on an agar plate will generate what we call a colony, so one of these dots. Okay? So by counting, those become visible to us, so now we can count them. So by counting the number of colonies and multiplying by the dilution factor, we know how many live cells there were in the initial product. Obviously, that cells cannot multiply and cannot produce colonies. This is why with this method, we can only count live cells, which can be a pro or a con, depending on what you want to do with those data. So that's also the reason why we need to dilute, because we could plate the undiluted, but then we would have, the plate would be so crowded with colonies that we would not be able to count it. Yes?
When we see the yogurt packaging, it does say best before. Is it because then it's best before that date because the number of like probiotic cells would be decreased so that it would no longer be best?
So, good question. So if it's a regular yogurt, no. Because in a regular yogurt, there are no probiotics, right? It's a normal yogurt. So the best before date has to take into account the time of spoilage, right? Because we want the product to be safe for consumption, so it doesn't have to spoil. So regular yogurts also contain some live cells, and which are living in your yogurt. So throughout the shelf life of the product, they modify the structure and the characteristic of the yogurt. So beyond the expiry date, it may be not necessarily unsafe to eat, it depends, but it may be unpleasant, so it may be spoiled. With probiotics yogurt, which are yogurts to which on top we add that microorganism that we have isolated and we know it's beneficial, and so we want to add it to the yogurt. So with probiotics yogurt, there is a specific regulation. So the actual probiotic microorganism that is added has to be there in a certain number until the end of the shelf life. So we want to be sure. Why is that? Because the studies that back up the addition of that product, so the research that has been conducted to support the benefits of that probiotic, were done with a certain number of cells, right? So we know that it works with that bacterial load. If it's less, we don't know. Right? So we want to be sure that what the consumer gets is what the science has shown to be effective. Right? So we want to be sure if it's 10 to the 9 cells per gram of yogurt that was tested in the clinical trial, we need to be sure that that's the amount that's present in the yogurt. So, you know, during the shelf life of the yogurt, the bacteria tend to die, right? Because when it's produced, they are in their best active habitat and form, and they're very active and they're doing well. And then they sit in a yogurt container for 2 or 3 weeks in a fridge, right? So, and they don't have like a continuous supply of nutrients and things like that. So their number, they die during the shelf life and their number decline. So this usually when probiotic producers put much more cells than expected in their yogurt, so that by the time it reaches the end of the shelf life, you hit the number that is supposed to be there based on the regulation. Yeah. Okay, so yes, so that's one of the methods we could use to count live cells. So it's largely used for food products, but it also can be used for stools to determine how many live bacteria of a certain type there are in the stool. So what could be a drawback of this methodology? So see, they have to grow on the agar plate. That means that we need to know what the bacterium that we want to count, what it likes to eat, like the type of food and nutrients and micronutrients that it needs. So with this methodology, we can count microorganisms that we know. Like we know what's their favorite carbohydrate source, what's their fermentation patterns, in which temperature and atmosphere we can grow them. So traditionally, this method was not usable for stool sample because until years ago, we were totally, like, we didn't have any knowledge of what most of the microorganisms in our intestinal tract would want to eat in a lab setting if we wanted to count them, right? So for the major species, the most represented one, yes, but there are more than 1,000 different species in the intestinal tract, and we don't know what's the optimal nutritional and growth profile for each of them, right? So this method was, as such, is not applicable in that context if we want to count bacteria that we don't know well enough. Though, as we will learn in 3 weeks, this method, and I don't want to tell everything now, but there is a new method which is called culturonics that builds on this, so on culturing, and that has been implemented to grow as many different bacteria as possible from the stools. So there is an adaptation of this for the stools, and we learn more about it in 3 lectures from now. Other methods to count, turbidity measurement, because bacterial cells scatter light in a way that is proportional to their number, so we can measure the optical density of a suspension of bacterial cells, and we can measure the dry weight of a cell pellet and in culture and determine their numbers as well. Okay, so bacteria, as you probably know, can be broadly classified into 2 main groups, the gram-positives and the gram-negatives. So I'm giving you a few details about this because this is the cell wall, the component of the cell wall of the bacteria are those that come into contact or in interaction, they interact with the host cells. So it's important to know what the cell wall of the bacteria contains in order to understand how our intestinal cells and cells of the immune system, how do they respond when they encounter the bacterial cells. So a main difference between the gram-positive and gram-negative cell wall is the thickness. So as it says here, the gram-positive can have up to 80 nanometer thick cell wall, while in the gram-negative is about 10 nanometers. So this is the representation of their structure. So as you can see for the gram-positive, there are multiple layers that are of peptidoglycan, which is, for bacteria, it's very typical because it contains murine, which we will see in a few slides what it is. And that is a distinguished distinguishing character of the bacterial cell wall. So peptidoglycans that are interspersed with, perpendicularly with these molecules that are called teichoic acids. On the other hand, the gram-negative cell wall has a much thinner peptidoglycan sheet of the membrane, and it contains an outer membrane that is attached to this peptidoglycan sheet. So pay attention to this molecule that is called the lipopolysaccharide, which is present only in the gram-negative bacteria, okay? And which is an inflammatory molecule that we will hear a lot during the next lectures. Yes?
Is the peptidoglycan sheet the cell wall? Is it
It's a
synonymous?
component of the cell wall. So all of this is the cell wall.
Oh, is the orange beige block, is that the inside of the cell?
Let me see if it's all
the
Yes, so we
rectangle?
have the membrane that covers the cytoplasm, and then we have the wall, which is the peptidoglycan sheets with the teichoic acids interspersed.
Can you repeat what you were saying about the inflammatory?
Yes, so in the gram-negatives, but not in the gram-positives, there is a molecule that is called lipopolysaccharide here in the membrane. And that lipopolysaccharide is a pro-inflammatory molecule. So both in the context of acute and chronic inflammation. So for example, in obesity, which is an inflammatory disease, many patients with obesity have increased concentrations of lipopolysaccharide in their blood, systemic, circulating lipopolysaccharide, and that lipopolysaccharide largely derives from the intestinal tract, from the bacteria in the intestinal tract. Can also come from the diet. So based on the structure of their cell wall, these bacteria can be distinguished in the lab because with the colorimetric assay, because they retain differently or not a dye which is called the crystal violet. So this was discovered in the 1800s by Graham, and the staining has taken his name, so Graham staining, and it's something that we can do very easily in the lab using this stain to just distinguish if what we are working at is a gram-positive or a gram-negative. So in our, yes.
So is it the peptidoglycan that causes it to retain the dye?
Yes, so it's the structure of the peptidoglycan, yes, because in the gram-negative is more loose, like there is less of this peptidoglycan sheet and is not able to retain the color, the dye.
So if you had like gram-negative and you put a lot of dye in it, would it be retained to some degree?
No, eventually no, because in the procedure you have there are multiple steps, which includes washing in the end. Otherwise, they will all look the same. So when you wash, then it would be washed away. So this slide shows. Don't be scared. You don't have to learn these structures, but I want you to have an idea of what they look like. So these are the lipoteichoic acids that run perpendicularly to the peptidoglycan sheets in the gram-positive cell wall. So it's just to show you that there can be of many different kinds. There are polymers of polyglycerol and polyribitol, and they also are substituted with amino acid and sugars. You don't have by any means to remember this structure, but I want you to know what they look like, and also to appreciate how there can be important differences between the different bacteria. So these are components of the cell wall. So every time that one of our immune cells, or intestinal cells, comes into contact with one of these bacteria, it has to be able to distinguish different types of molecules, right? Because, for example, the Clostridium inoculum would have type 3 LTA, and Staphylococcus aureus, which is a pathogen, has a type 1 LTA, right? So our cells have developed mechanism to distinguish between them and respond differently as necessary. Okay, so going back just a little bit to the peptidoglycan, because it's very important that you understand what it is, right? So this is an example of a peptidoglycan from E. coli, which is a gram-negative bacterium name to be remembered. You probably already know this name, and many of you may associate this name to a pathogenic bacterium, because I used to think of E. coli as the cause of enteric disease. But there are many, many different strains of E. coli, and all of us have E. coli in our intestinal tract, and we are perfectly fine, right? Some of these E. coli in our intestinal tract, we even need them. Some of them produce vitamins, for example. So not every E. coli is a pathogenic bacterium. Okay, so what does the peptidoglycan in E. coli looks like? And it's an easy one, so it's easy to look at the one in E. coli to better understand. So it is a polymer that contains a glycan backbone. So the glycan chain is made up of two sugars, the N-acetylglucosamine and the N-acetylmuramic acid. So the N-acetylmuramic acid is what constitutes, is the distinguishing character that I was telling you earlier, like the murine in the bacteria. It's only found in the bacteria. For example, the archaea, which are also unicellular, they don't have an acetylmuramic acid in their peptidoglycans. Okay, then the N-acetylmuramic acid is substituted here with with a peptidic chain that consists of 4 amino acids, which are this one listed here. Okay. These 4 amino acids, this chain, peptide chain, is what is cross-linked. So, as it's shown in C, the alanine at the end of one molecule is linked to the aminopimenic acid of the neighbor molecule, and with this interpeptide bond, it creates the cross-linking, and the polymer of the peptidoglycan is formed. Okay, a difference between gram-negatives and gram-positive that is important to remember, because later when we will have Dr. Philpott coming to the course talking about how the host handles all of these microorganisms, and when we need to understand what happens in this biotic situation, for example, inflammatory bowel disease, we will talk about the node receptors, which are receptors on the host side. So this receptor recognizes parts of the peptidoglycan molecules, and they can distinguish between gram-negatives and gram-positives. So for example, the NOD1 receptor recognizes what is in this black frame. So it recognizes that structure, including the mesodiaminopimenic acid. On the other hand, the NOD2 receptor only recognizes up to the glutamic acid. So this means that NOD1 recognizes the structures which is present in all the gram-negative bacteria and the gram-positive rod-shaped bacteria. So it's the minimal motif that is present in all of the peptidoglycans type. So it serves more as a general sensor. On the other hand, the NOD1 only can recognize with the mesodiaminopimenic acid, which is only present in the gram-negative. So these two receptors, NOD1 and 2, are part of our innate immune system and our innate immune response.
Sorry, which one was the general one? NOD2?
So the NOD2 is the general because it recognizes a motif that is present in all of them. Another component of the innate immune system is lysozyme, which we produce and which is present in the secretions like serum, tears, saliva, also in the intestinal tract. So what the lysozyme does, it cleaves the linkage, which is a beta-1,4 glycosidic linkage between the two sugars in the peptidoglycans. Right? So it cleaves this linkage here, the lysozyme. So it's part of our innate defense system because obviously by cleaving that you are leading, this leads to the ultimately to the death of the bacteria. Yeasts and molds. So very briefly, they are uni or multicellulars. They can reproduce by fission, budding, or sporulation. The one shown here, this is Saccharomyces cerevisiae, largely utilized in the food industry for baking products and also for alcoholic beverages. And the Penicillium roqueforti is common, is for production of cheese, but not all cheeses, like special, like for example French cheeses. These cheeses that are inoculated at a certain stage for their specific characteristics in the structure and taste. So that's what they look like. They also have their cell wall with the chitin, which is, so it contains a similar sugar to the peptidoglycan, the N-acetylglucosamine, but in this case it's a polymer of this sugar and glucan, which can be beta-1,3 or beta-1,6 linkages. And the differences between the yeast and the fungi is that in the yeast, on the outside, on the external side of this layer, we can have a monosylated proteins. Otherwise, they both contain this chitin, which is the polymer of this acetylglucosamine. Okay, now we move into the bacterial metabolism and basically the fermentation process and a little bit of respiration and anaerobic respiration. So if based on their metabolisms, the bacteria can be classified in three main groups. So the heterotrophs, they obtain energy from the oxidation of an organic compound. The autotrophs use an inorganic compound as a carbon or an energy source, like for example the methanobromide mediae, and the phototrophs, like the cyanobacteria, use light as an energy source. So let's see a little bit more in details what the heterotrophs do. So they use an organic compound, which can be a lipid, a protein, or a carbohydrate, as a carbon and energy source. And the process can be either the fermentation process or respiration. With fermentation, the energy is produced from the partial oxidation of the organic compound, and it uses organic intermediates as electron donors and acceptors. And respiration, as you know, it's the complete oxidation of the substrate by an outside electron acceptor, which can be oxygen, or in the case of anaerobic respiration, can be another inorganic or organic compound. So for the fermentation, which is the partial oxidation of an organic compound, that's the process, right, which I know that you're familiar with. So it leads, it's the glycolytic process, which in bacteria can happen through three different routes. The Enden-Meyer pathway, the phosphoketolase, or the Ender-Dudorov. Now, I'm going to give you only some information about the first two, which is what is mainly discussed and used for the production of the food products and also for what happens in the intestinal tract. So this glycolytic pathway produces the intermediate, which is pyruvate, right? And then these intermediates needs to be reduced in order to regenerate the NAD, right? So when it's reduced, these reduced end products are those metabolites that we keep talking about, that are produced by the bacterial fermentation, and which, once they are produced, they are available in the food product. They contribute to its taste, for example, or in the intestinal tract, they are available to us for absorption. And they can be beneficial or they can be detrimental. Okay, so two possible glycolytic pathways leading to different end products. Okay, so it's important to know this. The first one is the Ender-Meyerhof, which is probably one that you remember from your biochemistry courses. So I am not going through this because I assume that you know this, and there will not be questions on these reactions, right? But so just remember that from one molecule of glucose, we end up with two pyruvic acids because there is that split here into the two molecules of the glyceraldehyde triphosphate. Okay, so this pathway is the one that is used by the Saccharomyces cerevisiae that I mentioned earlier, and it is used by the homolactic bacteria, which are used for the production of many dairy products. Okay, so once we obtained the pyruvic acid, which is the oxidized intermediate, this pyruvic acid needs to be reduced, right, so that the NAD can be regenerated. So there are many ways that this can happen. So the ways that the pyruvate is reduced lead to many different end products, and different bacteria reduce the pyruvate in different manners and therefore produce different metabolites. So the easiest is the production of lactic acid in the lactic acid bacteria. So what do? All of the pyruvate that is produced is reduced to lactic acid. This is called lactic acid fermentation. So lactose glycolysis, pyruvate is produced, and then they have this enzyme, lactate dehydrogenase, that produces lactate. So it's the only product of their fermentation in this bacteria, in this homolactic fermentation, is the lactic acid. Other bacteria have much more complex fermentation processes that lead to many more products being produced even by the same bacteria, right? So for example, the Enterobacteriaceae, like E. coli, Salmonella, many of those enteric pathogens, they reduce the pyruvate to many products, including formic acid, and if they have the enzyme from formic acid, this can lead to the gases formation, hydrogen and carbon dioxide, and they can produce, if they are in the Clostridia, they can go down and produce acetic acid and isopropanol and butyric acid. Yes?
Just to clarify, for the lactic acid fermentation, that is the only one where the end product is the sole product of
Yes,
the...
if it is,
Okay.
so if it is Ender-Meyerhof pathway, then the pyruvate produces only lactic acid, and it's called homolactic fermentation. Yes, only lactic acid.
Okay, and in this page, all the different pathways that are shown, they're all Ender-Meyerhof.
Yes, they are all Ender-Meyerhof, so
Okay.
they all come from this, right? All have this, and once they produce the pyruvate, then the different bacteria have, it can go down multiple different routes, and different bacteria have different ways of reducing the pyruvate. So some of those options are shown here. So for example, when it's a homolactic bacterium, they produce only lactic acid from the pyruvate. On the other hand, Clostridia, from that pyruvate, they produce acetic acid, they produce isopropanol, butyric acid. The Enterobacteria, they produce formic acid, CO2, and hydrogen. And The bifidobacterium and the propionibacterium, on the other hand, go down this way and produce propionic acid and succinic acid. The other one written in blue there, like Klebsiella, Enterobacter, Bacillus, they go down on the right and produce those other products that are written there. Yeah, okay.
So which contains the different pathways to bacterium, you know,
It's how they have evolved and which enzymes they possess.
so for example, the same for bacteria, specific bacteria can use, well, I'm sorry, leads to two different pathways.
So for example, a lactic acid bacterium with homolactic fermentation, it can only do that one.
Okay.
So that bacterium, like Streptococcus, for example, it cannot produce isopropanol because it just doesn't have those enzymes. The Clostridia are also called the masters of the fermentation. They are very active fermenters. They can produce lots of different metabolites. But that's because their genome, it's large, it contains many more genes that encodes for all those enzymes that are needed for the production. For example, even the Enterobacteriaceae, like E. coli, Salmonella, that produce formic acid, not all of them can produce carbon dioxide and hydrogen for the formic acid. Only those that have the formate hydrogenase. So that's what it depends on. Yeah, there was a question there, and then go back to it. Yeah.
Are some of the substrates on this chart also found in the Krebs cycle?
Yes, like the succinic acid, for
Yeah.
example. Yes, it's the same
So-
one.
But these aren't part of the Krebs cycle?
No.
Okay.
Not
Maybe-
yet.
Just want to ask, like, when the bacteria convert the pyruvic acid and other things, is it for, like, do they use the end product for different functions, or is pyruvic acid
Not
like?
necessarily. So the main reason to do this is because they need to regenerate the nut. Because here, they need the nut so that, they need
Even
the
Krebs cycle.
But those metabolites may end up being useful for other microorganisms. For example, that would be the syntropy, right? So for example, those gases are used by the methanobrate bacteria. The lactic acid in the gut, the lactic acid, okay, the food products is necessary to make yogurt, we need the production of lactic acid, but in the gut, when the lactic acid bacteria produce lactic acid, then there is another bacterium which is called Veillonella that can use it, right? So the actual bacterium does it for its own good to regenerate its own mud, and then but it turns out that then the metabolites can be utilized as organic substrates by other bacteria. Sometimes they also do it because they need to detoxify. So I'm not showing this here because I would have to show you like hundreds of biochemical pathways. But sometimes a microorganism needs to detoxify. Maybe there is, like it happens to us, right? That we need to get rid of toxic products and we have the enteropathic circulation and the liver detoxifying. So sometimes they have to produce metabolites to get rid of toxic molecules that they're found in their environment.
Are we, for the test, should we try to memorize which bacteria does which pathway in, like, shown here?
So
or is that-
for the test, you have to remember the following that I was getting to. So yes, for the lactic acid, you must know which bacteria. There is another slide coming up with the heterolactic fermentation, and so you need to know the difference between homo and hetero, which bacteria do which one. But by which bacteria I mean this, right? Not remembering 50 different names, but just those three general that I put there and the one that we follow for the heterolactic. You will also hear it next week because the homolactic and the heterolactic fermentation are utilized for the production of dairy products. So Dr. Taimi will give you a few examples of few dairy products and which bacterium is used for those, and those are the ones that you have to remember. For this slide, what's important to remember that I was just going to mention is that the propionic acid, the butyric acid from the Clostridia, and the acetic acid, these are the short-chain fatty acids. So we will hear his name hundreds of times during this course. So they are here, and for these ones, yes, you have to remember, you have to know that Clostridium will produce butyric acid, but you know, you will hear it so many times in this course that I think you will not even have to study that. You will just remember just by coming to class. But you need to know the structures for these two. Like you need to know what they are.
Need to know the structure or don't?
Yes, like this one, like unit. Right, so because the short chain fatty acids, we must know what they are. They come up throughout the entire course from lecture 1 to the last lecture. Okay, so but just these three. You don't need to remember the structures or for the butanediol and the butanol. No, I will not ask that. But the short chain fatty acid, you need to know where they come from. Okay, so lactic acid, short chain fatty acids, and obviously you know now how the gases are produced in the intestinal tract and also in food products. Like sometimes we have the CO2 produced, like here for example, the propionibacterium produces propionic acid and releases carbon dioxide. And this is utilized for, you will see it next week, but in the production of Swiss cheese, those holes are created by the release of the CO2 during the fermentation of the milk for the production of the Swiss cheese. That's why it has holes. Okay. The heterolactic pathway does not use the enden layer, right? So remember we started here. So we have the glycolysis and we said it can happen through the enden-Meyer pathway, which is what we just discussed, or it can happen through the heterolactic pathway. So when it happens through the heterolactic pathways, we don't have two pyruvates that are produced. We only have one pyruvate, right? Because then there is a CO2 molecule that is released before the split. So after the split, we have one pyruvic acid that, same as to the homolactic fermentation, generates a lactic acid molecule. But from the acetyl phosphate, we get ethanol. Yes?
I'm sorry for like the bacteria in the slide just now that was like the streptococcus, lactobacillus, and there was one more.
Lactococcus, or I don't remember which one.
Yeah,
it was.
staphylococcus.
Oh, staphylococcus.
Are
Okay.
those all like homolactic or heterolactic?
Homo.
Okay.
Because they use the enden-Meyer and they produce only lactic acid.
Okay.
Now these ones, they do not use the enden-Meyer, they use the phosphoketoase or heterolactic pathway. And they produce one lactic acid because they only had one pyruvate. One ethanol, one carbon dioxide, which is released before the split, and energy. So these heterolactic bacteria, for example, some of the lactobacilli, so yes, so some lactobacilli use the homolactic and some other use the heterolactic. And these are, for example, the ones that I use to make kefir. You know that kefir, for those who have tried kefir, it has bubbles, it has this frizzy, right? So that's because of the CO2 that is produced in the heterolactic membrane. And leuconostoc, which is like a major genus utilized for making kefir. Okay, so this is for the key information that I wanted you to know about fermentation. For respiration, especially the aerobic respiration, I'm not going to tell you what respiration is, because I'm sure you know this, but I wanted to mention the anaerobic respiration. So the electron acceptors for respiration, or methanogenesis, which is an example of anaerobic respiration, right? So oxygen leading to water in the aerobic respiration, some Escherichia coli, for example, can do that. And I know that we have Escherichia coli also in the fermentation pathway, but that's because Escherichia coli is a facultative anaerobe, so it can adapt is its metabolism depending on the environmental conditions. On the other hand, a Methanococcus or a Methanobacter cannot adapt. They are strict anaerobe, so they would die even by super brief exposure to oxygen. This makes it very difficult to study them in the lab because it's hard to maintain a strict anaerobic environment. So if you remember from one of the first slides where I showed the Cristensenella minuta with the archaea, you may have noticed that there were some special tubes in that picture. Those tubes are not the regular ones. Those are tubes that are able to maintain an aerobic environment inside. So when we want to do those experiments, we have to order these special tubes if we want to grow the archaea. Another way in the lab is to have what is called an anaerobic chamber, which is like an enclosed space where we can choose the composition of the atmosphere inside this chamber. And so we can make it completely anoxic with no oxygen, and we can grow archaea. But this is not something that is very common. Like it's not every lab can do this. So it's hard to grow and study archaea in the lab, as opposed to E. coli, which is one of the easiest that we can have to study. Okay, so these are just examples of electron acceptor, and I want you to remember this one, right? So the CO2 for the methanogenesis reaction leading to the production of the methane. So for this, you have to know the reaction of methanogenesis that is performed by the methanogenic archaea, not by bacteria. Only archaea can do this. So hydrogen and CO2 that derive from the bacteria, right, as we have seen in the case of the gut ecosystem, leading to the production of methane, water, and energy. So, did I already say everything about this slide? I think so. Yes? Okay. So the last thing that I wanted to mention today is the construction and utilization of phylogenetic tree. And first of all, I wanted to situate what we have been talking about, so bacteria and archaea versus the eukaryotes on the tree of life, right? And so, as you can see, they are different domains. So the archaea are not bacteria, right? So I'm stressing this because until 25 years ago, we thought that archaea were bacteria, right? So, and they were called archaeobacteria, right? But now we know that they are a totally separate domain, so it's the three different ones. So you may come across these phylogenetic trees in the literature and in the papers and maybe in some of the lectures. So they are used to understand the relationship between the different microorganisms. And what we are using for this is the sequence of the 16S ribosomal RNA in these microorganisms. So based on the sequencing of the 16S RNA, we can determine the degree of relatedness between the different species. So it gives me also the opportunity to mention how these bacteria and archaea are classified, right? So I know that you know the taxonomy is important. So when we look at the phylogenetic tree, the highest taxon, so the less specific, is the phylum. Sometimes it used to be called the domain, but domain should be even on top of the phylum, right? So when we will talk about the composition of intestinal microbiota, for example, we will say, oh, today we will talk about 4 phyla in the intestinal tract. So that's what we mean. So phylum is the highest taxon within a domain. So it's a Latin word. So the plural is phyla, right? So there is a lot of Latin in this microorganism's name. So the taxonomic ranks below phylum are those ones there: class, order, family, genus, and species. When we provide the name of a microorganisms, right, like E. coli, Escherichia coli, this is a binomial that contains the genus and the species Right? So Escherichia is the genus, coli is the species. And they are written in italics. So it must be written in italics. So in your assignments, please, written assignment, the summary, and the final cumulative assessment. Every time you write a microorganism name, this has to be in italics, if it is genus and species. The domain does not need to be in italics, but the name of the bacterium, yes. So don't forget this. Okay, so I mentioned the 16S RNA. That's because that's the RNA molecule in the subunits of the prokaryotes, right? So this 16S RNA is very useful for us phylogenetically because it contains conserved and variable. Yeah.
Just asking, does it have to be started with capital letter, the bacteria?
The genus, yes, but the species, no.
Oh.
Like it is here. So we use the 16S RNA for phylogenetic studies, and we exploit this phylogenetic knowledge, for example, to study the composition of a bacterial community, which can be in a food, it can be in the stool of a person. So that's a gene that we use a lot, and that we will talk about 16S based data during this course. So I just wanted to give like a little refresher that this molecule contains variable and conserved regions. So these variables and conserved regions, so for example here there is a picture for E. coli and the 9 different variable regions in its 16S RNA molecules. So obviously the conserved regions are conserved phylogenetically across evolution species, and the variable regions are the one that we use to distinguish among different bacteria, for example. So we have like a complex sample, like a stool sample, and we want to know which bacteria are there, but we don't know how to grow them, we don't have any knowledge about what's present. So what do? What we can do is to sequence the 16S RNA in that sample, and then based on our databases and how the different variable region sequences align, we can determine its composition. So this is what is typically used to construct the phylogenetic trees. So it's a small gene, so it's easy to sequence in this experiment. Like we don't have to sequence too long. So we will have lots of 16S data that we'll talk about during the course. So this is all that I had for today. Is it clear to you, or you have any questions remaining? Yes.
Would you be able to go over the heterolactic pathway slide coursework?
Again, like repeat the whole thing.
Sorry.
Okay. So in the heterolactic pathway is it's an alternative glycolytic pathway, right? Up to here, from glucose to pyruvic acid, right? So in the Mayer-Hoff, we know, right? I think you remember from the glucose, there is no CO2 produced, right? You have the phosphate, it gets phosphorylated, and at the split, there are 2 equal arms because there are 2 3-carbon molecules, 2 molecules of the glyceraldehyde 3-phosphate on both sides, and both lead to pyruvic acid being produced. So in the case of the homolactic, we have 1 glucose and 2 lactic acid. In the heterolactic, it's a different enzymes here that these bacteria have, right? So there is the release of a carbon already there that produces the carbon dioxide, which leaves us with the 5 carbon molecules that now is split into 3 and 2. So the 3 is the same as the Mayer-Hoff. It's a glyceraldehyde 3-phosphate that leads to pyruvic acid. And on the other side, the acetyl phosphate that leads to ethanol. Okay? once we have the one pyruvic acid on this side, again, we have to regenerate the NAD, right? And in the heterolactic fermentation, we do this, those bacteria do this by producing lactic acid, which is the reduced end product of the heterolactic fermentation. But there is only one lactic acid, right? So then there is one CO2 that comes from here, the carboxylation before the split, and then there is one ethanol from the other arm. So it's called heterolactic because there are different products being formed here. Is it clear? Yeah.
So the homolactic fermentation has produced two lactic from two pyruvate?
The homolactic produces only lactic acid.
Oh, yeah.
But because there are two molecules of pyruvate, right, because the split is equal 3 and 3, then each of those generates one lactic acid each. So two lactic acids, so only lactic acid ultimately in the homolactic. Yeah? Yeah?
Is it only is it only heterolactic bacteria that use this sort of like the what's it called? Phosphatidylase.
Yes,
acid,
yes.
Is it like as I understand, the phosphatidylase makes one pyruvic acid, and then that pyruvic acid is further reduced to lactic acid by heterolactic bacteria. That phosphatidylase pathway is it only used for heterolactic bacteria, or are there other bacteria that use it but don't reduce it to lactic acid?
No. Heterolactic includes the whole thing.
Oh, okay.
Because those heterolactic bacteria, they cannot just only do this, right? Because the way they obtain the pyruvic acid is through the phosphatidylase.
No, I meant like, are there any bacteria that use only the phosphatidylase but use the N-pyruvic acid to form like something else, another N?
I don't think so.
Oh, okay.
I don't think so. No, so we have an easier outcome here. We don't have all those complex metabolites as we have them there.
For the ended fermentation, I was wondering the E. coli and the salmonella, do they reduce the formic acid and the oxalic acid, or is it?
The formic acid and the
The E. coli and the salmonella, do they reduce both the formic acid and the oxalic acid?
So, yeah, it depends. Some of them, if they have the formates, dehydrogenase, they produce the gases, not all.
So they mainly, which one do they mainly reduce? Do they mainly reduce the oxalic acid? Or, like I'm trying to understand what role do the E. coli and the salmonella play?
Yes, so it's difficult to, some E. coli will do up to the formic acid, and some will do to the CO2 and the hydrogen depending, right? So you just can remember that the Enterobacteriaceae will produce formic acid and may produce CO2 and hydrogen. Because it's difficult to group them, right? It's also for the Clostridium. There will be other bacteria that are not here that produce butyric acid, right? But this is like a general view to give you an idea of what's going on. Otherwise, it's impossible to go into the details of every single bacterium. So it would be too much. It would be too overwhelming, right? So, but it's not, I realize it's not like a perfect summary because it's just impossible to give it. Some of them also, they produce different products, like the same bacterium in different situations, right? So it makes it, it depends maybe on the substrate that it has until how far it goes down into the reactions. So it's difficult to give a definitive answer. It's much easier for the homolactic because they just produce that and that's easier. But the enterobacteria are more complicated and the Clostridium even more. Right? There is many more products that they make beyond these. So they, like the vitamins that I mentioned, for example, there is much more. So I just tried to group them to make it understandable and give you an idea of what's going on. Yes.
Which lifestyle pathway is more prevalent?
Which is more prevalent?
Yeah.
In which sense?
Like within like the intestinal tract?
Oh, It's hard to answer. I think I would say not the homolactic, right? Endomayo probably. But again, it depends on which bacteria are present and which genes they would express at that time, right? But I would say like this side of the slide. The homolactic, yes, there is lactic acid produced in the intestinal tract, but this is largely happening in food products. Right? In the gut, this part of the slide, it's very complex, the fermentation in the gut, and it's mixed acid fermentation, which many different products produce, especially because the lactobacilli are not a main genus in the gut. On the other hand, Clostridia, it's one of the main. It's also the most abundant. So there is more of that part of the slide in the gut.
Just want to ask, like for the task, do we have to remember like which bacteria go for which pathway in general in which product? Like do we get a question like that?
So what I said earlier, right? So for the homolactic, yes, and especially those that are mentioned next week for specific products.
Specific.
Right, so you will hear next week for making yogurt, we need these. So these are the ones that you need to remember. For this slide, you need to remember which produce short-chain fatty acid. For example, now you know that the Clostridia are like a large producer of short-chain fatty acids, for example, right? But I will not ask you what does Shigella produces. Like there will not be questions like this. Mainly this knowledge you needed to understand what we'll discuss in the next lecture. So if I tell you short-chain fatty acids are absorbed and the intestinal cells rely on butyric acid as their main energy source, you know where it's coming from. It's from the bacterial fermentation and it's mainly the Clostridia. But I will not put questions in the midterm, what does Klebsiella produces.
And for the gram-positive bacteria, for the teichoic acid, that's also is it the same as like teichoic acid as LTA?
So LTA is the acronym for lipoteichoic acid.
So is it the same as the teichoic acid?
LTA is the teichoic acid, not the lipopolysaccharide.
That's only for gram-positive.
Yes, the LTA is for, the LTA is for the gram-positives.
Okay. And for the muriatic acid, is it only for the
No, that is in all. The murine,
Yeah.
no, that's in all bacteria.
Oh, so even gram-negative has muriatic
Yes,
in the pedicle.
yes, but it's a much thinner layer.
Okay.
Sorry, I have another one. For lactobacillus, I see it here and also the Catarrhactis, so it's like different strain, has different some
Species.
homo and some
Species.
some species.
Yeah.
There's some homo and some hetero.
Yes. Good. If you have more questions, you can ask next week or you can